All right, sorry to have missed your opening comments, Joe, on that. Let's see, I have to do continue. So thank you, Joe, for those welcoming remarks. And I want to add my thanks on behalf of University Relations Council to Janet Berry, our good friend and ARCS member, and Jessica Farler-Ward, who did such an amazing job in working with our team, Connie and Melanie, to put this together. And also to Dean DePlanck. Your generous and really tremendous partnership with ARCS is above and beyond, and all of your efforts in putting today together, especially given the wins and the lack of potential power. It is a great honor now to recognize the ARCS scholars for their accomplishments and their selections by UC Davis as recipients of the ARCS Award. So I want to speak directly to the ARCS scholars. We are thrilled to have you with us today, and as Joe mentioned, to celebrate you. And it's great to see, well, I actually not sure I can see all of them, but many of you received the medallions and the certificates. I think others are in the mail because of your various locations, but it's just a tangible representation of how proud we are of you. And especially during this time of physical distance, when we can't get together other than across this glass screen, we're just really, really wanting you to know how proud we are of you and how important your association with ARCS is. So as members of the 2020-21 cohort, you are 18 of the 79 science, engineering, and medical research scholars in Northern California to be selected as ARCS scholars this year. And UC Davis is one, as you may know, of 6 university partners to our chapter. And through those 6 universities, we've awarded this award 2,800 and more times. Some recipients multiple years, but that's how many times we've given this award. And as you may have heard, we are now celebrating our 50th anniversary this year. So it's a very special year for us to recognize you as ARCS scholars. And we welcome you into this community of excellence with those who have received this prestigious award around the country over the years. In fact, nearly 11,000 ARCS awards have been granted since our founding in 1958. And if you haven't heard the story of why and how ARCS came to be, do check out our website because it's a really good story. So today we salute you, and we look forward to hearing about each one of you and your research and what you're doing both in the labs and in the fields. This past year, as we all know all too well, has been profoundly challenging, shaking the norms of how we work and how we interact. And so we're especially applauding today your perseverance, your ingenuity, your resourcefulness. And we know that UC Davis, your professors, PIs, the dean, and everyone are doing everything they can to ensure the best possible support for you as you drive your discoveries. We want you to know that we're inspired by you, and we want you to know we are firmly in your corner. We believe in you, we're rooting for you, and not just now, but throughout the years at UC Davis and throughout your careers. Our support is more personal than just funding. And we want you to know that we truly care about your success. And we hope that the ARCS Award makes a big difference to you, just as you are making a huge difference to the world. And science, thanks goodness, is in the spotlight now more than ever. And you are the problem solvers that humanity and the planet truly need. So we hope you'll stay in touch and share your stories with us through the years ahead. We hope you'll stay connected with ARCS colleagues, with ARCS members and our friends and our donors through our website, through social media, and best of all in person. But back to the present for now, I'd like to have everyone come off mute, if you're on mute, for just a moment, and join us as we recognize with a round of applause this incredibly talented cohort of scholars full of brilliance and potential. And so now our virtual applause through the waves.
Hey.
Oh, like that. Woo, woo. Excellent. So truly, truly, we want to celebrate you today. And now it's my great pleasure to introduce Dean DePlanck, Vice Provost and Dean of Graduate Studies. And Dean DePlanck, widely known as JP, was appointed to this position exactly 2 years ago in January of 2019. And JP, you've been an incredible partner and friend to ARCS, and we're so impressed by your focus on cultivating an inclusive, equitable learning environment for graduate students and postdocs so that they are equipped to spearhead discoveries with global impact. We're in awe of how you teach mechanical and aerospace engineering, yes, truly a rocket scientist, conduct research, and handle all the administrative duties of your position. Clearly passionate about your work and smartly surrounded by great people. Your leadership is a joy to watch. Thank you for being with us this morning to introduce the 2020-21 ARC Scholars. And so now we turn the program to you and your outstanding UC Davis scientists.
Thank you. Thank you so much. Those are very kind words. I want to thank both you, Chris, and MJ Whitehouse for this welcome introduction. I mean, it's really great to see everyone again here today. The events that we have with ARCS, like the Frontiers of Science, the symposium over the years, I mean, I look forward to those always. The energy that it represents, and also the encapsulation of the support that for our scholars is amazing to me. And you're right. So I was appointed 2 years ago. Before that, I served as associate dean in graduate studies for graduate students and scholar. And already I had the opportunity to participate in the symposium, and that was my introduction. And I've enjoyed it tremendously since then. The ARCS Northern California chapter has been a long-standing partner for UC Davis. I mean, 49 years. 49 years. That means that next year we'll be celebrating a big anniversary, 50th year partnership. So just 1 year after your 50-year anniversary. I'm really hope that we'll be able to do that in our newly built home at the Graduate Center. So I'll just point it out again. It's not easy on Zoom, right? But if you look behind me, you can see it says Graduate Center here. If the picture is not too small, it's actually a picture of the building as it is now. We'll start moving boxes within a week, within less than a week. Of course, moving people in there is going to take a little bit longer, vaccinations permitting. But next year, we really hope that we'll be able to actually celebrate that 50th-year anniversary in the center. Through the dedication and commitment of ARCS, hundreds of scholars have been supported, more than $3.3 million of support, in fact. And we want to thank you for such a strong partnerships. Yes, thank you. Those are two very important words that they do come from our hearts. You're truly fulfilling the mission to advance science and technology in the US by supporting UC Davis graduate students and their ability to conduct innovative research. Today, we are proud to introduce and celebrate the 2020-2021 cohort of ARCS Fellows. So I think that almost all 18 scholars are here virtually today. The plan was for all of them to be here. We have a couple with power outage issues. So I think it's 2 or 3, not more. And I'll mention it as we go through the program. Some had anticipated possible challenges with internet connections, even before knowing that the storm was going to be here. And because they were still eager to share their stories with you, with us, they have a pre-recorded video that we'll be able to play. I think we have a couple of those. At the end of the program, you will have the opportunity to talk directly with the fellows in breakout rooms, if you so choose. This is our effort at trying to make it feel as close as possible to what would happen in person, right? Where you would go and be able to chat with the fellows if we were in person. Anyway, without further ado, let's turn our attention to the virtual spotlight to the scholars. I think the first, yes. So our first scholar is Anna Adhikari. She's a PhD candidate in molecular, cellular, and integrative physiology.
Thank you. Good morning, everyone. My name is Anna Adhikari. I'm a third-year graduate student in the Molecular, Cellular, and Integrative Physiology grad group. My research is in cell-based gene therapy, specifically leveraging blood stem cells to deliver therapeutics into brain in an attempt to develop a treatment for Angelman syndrome. Angelman syndrome is a rare neurodevelopmental disorder. It's caused, or it has been identified, the causal gene being a paternally imprinted UB3A in neurons. So our approach is to provide that missing protein, package it in a lentiviral vector transduced in blood stem cells, and deliver that missing protein into the neurons. Thank you so much to the ARCS Foundation and the sponsors for supporting scientists like me and our studies, building this wonderful, wonderful community and fostering student development. It's truly a privilege to be a part of this community. I'm very grateful. Thank you again.
Thank you, Anna. Our next scholar is Charlie Alex, PhD in Integrative Pathobiology.
Good morning. Thanks for the opportunity to share a little bit about the work, and thanks, of course, to the ARCS Foundation for your support. It's really really a huge help. So I'm a second-year student in integrative pathobiology, and my research is focused on persistent viral infections. So these are viruses that we and all animals are carrying around with us probably for life, and they can be asymptomatic or maybe only cause problems for us when our immune systems become compromised, or when they jump into a new host species, or maybe they're contributing to chronic progressive diseases, so that they're very difficult to actually attribute specific diseases to or consequences to. We don't have good lab animals for a lot of these viruses, and there are real limitations to how we can understand them from just purely epidemiology, which is how it's been approached pretty frequently. But we're starting to realize that these infections are really important, much more important than previously recognized. And so so my research is focused on trying to get an understanding of how these persistent infections work. And that's the context. In particular, I'm focused on a group of persistent parvoviruses that we discovered in red pandas. This is a discovery in our lab, and we found now that about half of red pandas in US zoos are carrying these infections, usually totally asymptomatically. But it's kind of ironically this zoo-housed population of this endangered species, this really carefully managed population, that gives us a real opportunity to understand the natural history of these viruses, how they're interacting with their hosts long term, because those populations are so carefully managed. So my program is called Integrative Pathobiology, and this is really an integrative approach. We're using things like phylogenetics and genetic characteristics of the viruses, epidemiological modeling to understand how infections may be transmitted through the population. I have a background as a veterinary pathologist, and so I'm using archived necropsy materials and molecular labeling methods to try to understand if and when these viruses can be associated with specific diseases. And we're also using kind of traditional classical virology methods in cell culture to see if we can predict when these viruses may be associated with increased virulence or potentially a spillover to novel host, which we know happens with some of these viruses. So that's the overview. Thanks again to the ARCS Foundation for your support. I appreciate it.
Thank you, Charlie. Our next caller is supposed to be Peter Andrew, PhD in pharmacology and toxicology. I believe his power is out. So I'm looking around in case he was able to come on board, but I don't see him. So we'll move on to the next caller. Next is Savannah Conan, PhD in chemistry. Savannah?
Hi everyone, I'm Savannah Conlon. Thank you for the introduction. I'm really excited to be here today to talk to you a little bit about my research and be a part of this community. A little bit about myself. I actually was first inspired to pursue chemistry from my high school chemistry teacher, Mrs. McDowell. I still remember her helping me apply for college and figure out what I want to do. And so I did end up attending college at UC Santa Cruz, where I worked with Dr. Scott Loki, and that's kind of where I discovered undergraduate research and the process of scientific research. And he really inspired me to pursue my PhD in chemistry. So here I am. I work with Dr. Sheila David in the Department of Chemistry at UC Davis. I'm a fourth-year graduate student. And in the David Lab, we study DNA damage, how it arises, and how it can be repaired from the genome. DNA damage that's left unrepaired can be absolutely detrimental to one's health. It can lead to a myriad of diseases, cancer, metabolic disorders, neurodegeneration. And so it's really important that we study and understand the repair pathways that allow us to prevent such diseases from causing problems. And so my specific research project, I'd love to talk to you more about in the breakout room. It's focused on a repair enzyme known as NEAL. So thanks to the ARCS Foundation, I've really been able to focus more on my research project this quarter, and especially during the midst of a global pandemic, to be able to provide such support to so many students, it cannot be thanked enough. So I look forward to speaking with you further.
Thank you, Savannah. Our next scholar is Katherine Corn, PhD in population biology.
Hi, I'm Katherine Corn. Thanks so much for having me here today, and thank you so much for your support, ARCS. So I'm a fifth-year PhD candidate at UC Davis, and this is my second year being supported as an ARCS scholar. Before coming to Davis, I did my undergraduate work at Cornell, and then most of my research at the University of Washington's Friday Harbor Marine Labs. My research focuses on the patterns and the processes that underlie the diversity of life on Earth. So I'm really interested in how evolution over millions of years produces the amazing array of animals that we have alive today. And so specifically, I study how the physical properties of interaction with the environment that surrounds an organism affects its evolution. And so I focus on feeding, which is a particularly crucial aspect of the animal's life. Without feeding, you don't get to do anything else. And I study how the different ways that fishes are able to capture their prey on coral reefs affects their diversity. So my research uses statistical modeling, three-dimensional high-speed videography, and the largest ever fish body shape data set to understand how the mechanics of feeding affects the macro scale evolutionary processes underlying the fabulous diversity of coral reef fishes. And I'm really, really grateful to the ARCS Foundation. ARCS has given me crucial support. It has paid my bills and allowed me to do the really time-intensive research that is crucial to working with large data sets. So thank you so much. And then I'll be in a breakout room later if you want to chat.
Thank you, Katherine. There's seemingly beautiful weather where you are. We're all envious.
Belize is very nice.
Our next scholar is Kailyn Dietrich, PhD in soils and biogeochemistry. And I think we have a video for Kailyn.
I do. Let me share my screen. Give me 1 second.
Thank you, Jessica.
Okay, I'm going to share my screen. Where did the video go? 1 second. Technical difficulties. There we go. Where is it going? Okay. Oh, this video keeps disappearing on me.
Oh,
Can we see it now?
Yep.
How's the sound? Did you hear the sound?
No.
No. Okay.
PhD candidate here at University of California, Davis. Originally from Northern Colorado, I got my Bachelor's of Science in Soil and Crop Science and Organic Agriculture at Colorado State University. From there, I moved to Madison, Wisconsin, where I got my master's degree in agroecology and soil science at UW-Madison. Now here at UC Davis, I am in the Soils and Biogeochemistry graduate group. My dissertation research largely focuses on a novel perennial grain crop known as kernza. Kernza is an agronomic crop that produces a wheat-like grain that we can use in cereal and beers and bread. However, unlike annual wheat, we don't have to till and replant kernza each year because it is a perennial. Most of the research prior to 2017 on kernza has taken place in the Midwest and Northeast USA. So in 2017, my advisors and I started the first long-term field experiment of kernza in the state of California, where we compare kernza to both tilled and no-till annual wheat along gradients of water, irrigation, and nitrogen fertilization. When I say compare, what I mean is that we are comparing the above ground productivity, so looking at biomass and grain yields, as well as below ground productivity, such as soil carbon sequestration, soil health, and the soil microbiome. First and foremost, we're hoping that the impact of this research is to inform Central Valley growers on whether kernza may or may not be appropriate for their specific agroecosystem, as well as more broadly trying to understand the underpinnings of soil ecosystem services and whether they're being driven by perennial root presence, a lack of soil disturbance, or both. I would like to sincerely thank ARCS for making my work possible here at UC Davis. I'm honored to be part of this community, and I'm immensely thankful for your support. Thank you.
Thank you, Kailyn. And Jessica. Our next scholar is Emmett Francis, PhD in biomedical engineering.
Hello. Yeah, can you hear me? Cool. Yeah, so hi, I'm Emmett Francis. I am a fourth-year PhD student in biomedical engineering. I do biophysics research in the Heinrich lab, and I'm actually third-year ARCS scholar, so thanks so much for the support over the years. Generally, our lab examines the physics behind how white blood cells chase after and engulf different pathogens, including bacteria, fungi, or even viruses, like we're also aware of right now. So I focus on a particular white blood cell called the neutrophil. It's the most abundant one, and it's the first one to respond when you get a wound or any sort of infection. In my PhD, I'm particularly thinking about how calcium inside of neutrophils plays an important signaling role during the process of engulfing pathogens, and that process is called phagocytosis. And in my experiments, I consistently observed this rapid burst of calcium concentration during phagocytosis. And while it's generally known that calcium is really important for cells to be able to send signals within themselves, it's not exactly known why this signal in particular is important for phagocytosis. We think it's important for how the cell is able to change shape and exert forces, but really narrowing that down is what I'm working on over the next year or so at this point. So thank you so much to ARCS for the support. It's been so helpful over the years.
Thank you, Emmett. Our next scholar is Hilary Green, a PhD in agricultural and environmental chemistry. Hilary?
Hello, it's so good to be here today. Thank you so much. I wanted to start with a little bit about myself. I've always been passionate about agriculture, food, and chemistry. And I once decided to find the best way to cook an egg. And in one of my trials, I put an egg in the oven at 140 degrees. And when I took it out, the yolk was almost like Play-Doh or silly putty. And this certainly wasn't a very appetizing result, but it was an interesting discovery. And it's that element of discovery that keeps me going in my research. So my expertise is in lipids, more specifically edible oils like olive oil. and each one has its own unique chemical makeup or fingerprint. However, for avocado oil specifically right now, there are no standards or there's no established fingerprint for it, which in essence means that anything can be poured into a bottle and labeled avocado oil, and we have no official regulations to determine if that's true or not. As you can imagine, that's a major risk for consumers and the growth of the industry. So one of my recent studies, which I was actually able to complete last year, thanks to funding from ARCS, I analyzed avocado oils currently sold on the market. And we found that there were multiple samples that were 100% soybean oil, others had different seed oils in them, and many of them were of poor quality. So this study, the purpose of that study was to serve as a call to action for the FDA to actually develop standards. So actually now what I'm doing is I am working with them to help understand that avocado oil fingerprint so that we can appropriately develop standards for avocado oil. And this is a little bit more complicated than it sounds. There's a lot of natural factors that play a role. It depends on the variety of avocados, the harvest season. And so I'm currently getting samples from multiple countries over multiple time points to try to do this. And the amount of samples needed for that kind of study isn't trivial. And so I thank ARCS again this year for their support in helping me continue to complete this research to keep consumers safe and informed, and also to hold industry accountable for their products.
Thank you, Hilary. Our next speaker is Michael Hugh in the PhD in geology.
Yep, thank you. My name is Michael Huff, and I am a geochemist that studies volcanic rocks from the bottom of the Atlantic Ocean. We know that hydrogen, carbon, and nitrogen are essential elements for life on Earth. However, what is unknown is when these elements were delivered to the Earth, the amount of these elements that were delivered, and if the surface conditions of the early Earth was conducive to the formation of the molecules that make up life. Clues to these answers are trapped within the Earth's interior. However, hydrogen, carbon, and nitrogen are reactive elements. Thus, they have lost their primordial signature in the rocks that are produced today. So instead, I measure the noble gases, which are inert and are excellent tracers of the components that delivered the essential elements for life. But the noble gases have their own set of challenges, primarily due to their extremely low abundances in the range of picomoles to femtomoles. I want to thank ARCS for their support, as their funding allows me to utilize the latest generation of mass spectrometers and procedures to make measurements that have historically been difficult and often impossible to make. Furthermore, I am proud to say that I am a father of a 1-year-old, and what I'll call COVID parenthood has added both financial and time management challenges. And the ARCS funding allows me to focus on what is important, my family and research. Thank you.
Thank you, Michael. Yeah, challenges I can imagine with a 1-year-old running around. My apologies also for mispronouncing your name. I kind of missed on that. Our next scholar is Zoe Canavas from a PhD in Civil and Environmental Engineering. I don't think Zoe is with us. Power out. Is that true? Yes. Okay. All right. Okay. And the next speaker is Kelsey Leiberger, PhD in Population Biology. Kelsey?
Hi, my name is Kelsey Leiberger, and I'm a 6th-year PhD candidate in population biology, and this is my 2nd year as an ARCS scholar, so I want to thank you so much for your support. I study the evolutionary and ecological effects of environmental disturbance, particularly in freshwater communities. And so whether this disturbance is because of an invasion of a new species or some sort of extreme event like hurricanes, droughts, and floods. And I study these with a variety of approaches, both manipulative experiments in the field, but also with theoretical modeling. So I look forward to discussing those with you in breakout rooms. So thank you again for your support in making my research possible.
Thank you, Kelsey. It's a live from the lab broadcast here. Our next scholar is Alex McIntyre, PhD in animal behavior. Is Alex with us? I have him as present, but
He's here.
Can you hear me better now?
yes.
Sorry, it's a math problem. Anyway, hello. Sorry, I'm also live from the lab. Thank you so much for organizing this. I'm really excited to be here today. I'm a fifth-year PhD candidate in animal behavior at UC Davis, and this is also my second time receiving the ARCS funding, which has been wonderful. I'm originally from Ohio, which may be surprising given that I'm now a marine biologist. And I did my undergrad degree at Williams College of Massachusetts, where I majored in biology and English. So my research focuses on how environmental variables affect movement, behavior, and survival in marine organisms like sharks and salmon. I primarily study shark behavior in the field, so I'm here to debunk anything you might think you know about Jaws. And for the last 3 years before COVID, I studied basking sharks in Ireland, focusing on their use of Irish waters and social behavior. And I do this primarily using electronic tags and videos, which the previous ARCS funding has supported. Again, unfortunately, this year that field season was canceled, but I am excited to be going to French Polynesia in the fall to study social behavior in juvenile blacktip reef sharks. But not all of my work is sharks. I'm currently in the lab and I'm looking at how predation risk of juvenile salmon increases with increasing temperature. And the spoiler alert is that we do see more salmon being eaten when temperatures rise, which might not bode well for your favorite food in light of climate change. I'm also really interested in science communication. So if you are interested in learning more about my work, I have a personal website you can Google, and you can also check out the Irish Basking Shark Group website, which is something that I'm a big part of. And I'm also happy to chat more about my work in breakout rooms. And I guess I want to conclude by, again, thanking the ARCS Foundation so much for making my research possible, especially in light of COVID. I've definitely hit some hiccups during my PhD. COVID is one of them. But also it's very hard to study large, elusive, endangered animals. And this funding has allowed me to do that. So I really wish that we could all have met at dinner this year. But I'm grateful to be meeting you all today on this platform.
Thank you, Alex. Yeah, I mean, the French Polynesia angle is kind of interesting, though. That's going to be an interesting place to do marine biology. The next scholar is Conor Meyer, in a PhD in biomedical engineering.
Are we having technical difficulties? I see him on. Maybe move on and we can come back. I'll try to send a message.
Yeah, we'll do that. Let's go to Emilia Monson, PhD in animal behavior.
Hi, yeah, my name is Emilia Monson. I'm a 6th-year PhD candidate in animal behavior. And for a long time, I have been really interested in this sort of big general question of how our experiences make us who we are, and specifically how the things we experience prepare us to deal with challenges that we might face. And so I work with tiny fish. I work with little tiny fish. And so their big challenge that they face are environmental pressures, but also predation challenges. So I've been doing a lot of research looking at how experience with predators, both during development and also as an adult, shapes the way that animals respond to general challenges that they face later on. So how does experiencing one type of predator when you're a baby affect your ability to respond to a novel predator that you've never had any experience with as an adult? I've also gotten, most of my research really looks at sort of the scary side of things, but recently I've gotten interested in thinking about what are the good things that we can experience that can make us better at dealing with challenges. So I've been looking at how experience with different types of social group affect the way that fish respond to challenges at later time points, which has been super fun and a little bit more upbeat than looking at constantly fish being predated upon. And I wanted to thank ARCS so sincerely. This is my second year being funded by them, and the ability, when the coronavirus hit originally and we went on lockdown, my research took a major hit. I was still able to go in and take care of my fish, but I couldn't do the research that I had planned, and I wasn't able to get new fish even once things sort of opened back up again because my fish sources are still on lockdown. So it was a huge blow to my PhD, but having the ARCS money and having the flexibility that it enabled allowed me to purchase software that I wouldn't have had access to otherwise, that enabled me to look at my existing data in different ways and kept me on track to graduate, which is phenomenal because I'm going to be graduating in June, where I'm starting my absolute dream postdoc in Scotland, doing research on temperature and predation effects on social behavior in minnows. And I'm so excited, and I definitely could not have gotten there without the ARC support. So the fact that you guys enabled that flexibility is just, keep doing what you're doing. It's incredibly helpful for us. So thank you so much.
Thank you, Emilia. And congratulations on the postdoc offer in Scotland. Sounds exciting. So I think Conor had sound issues, but those are fixed now. Can we go to Conor?
Yeah.
Okay.
Yeah, sorry about that. I was
okay.
yeah, just like cut out, and I was like, I don't know what's happening. Okay, well, hi everyone. My name is Connery. I'm a third-year PhD student in biomedical engineering. A little background, kind of growing up, I love building things out of anything I can get a hold of, usually like robotics or whatever else. Then I found out about genes, and I thought that I could just throw 20 different amino acids together and do virtually anything that nature can do. I was pretty hooked. And so since then, I haven't really stopped independent research to try and engineer and utilize these proteins. So my current project uses what are called like cell-free protein synthesis systems. And what these are essentially is just taking cells, popping them open, and harvesting their innards to essentially reconstitute cellular processes. So the main thing that I'm interested in is reconstituting protein synthesis. And so with this, now I can mix all this stuff in a test tube and produce proteins on demand. And so specifically what I'm looking at is producing membrane proteins inside these mixes. Membrane proteins have been notoriously difficult to make because inside cells, you need very specific machinery and chaperones and all that kind of stuff in lipid environments, and then extraction and resolubilization and detergents is super difficult. And so if you can make it in this highly composable environment, it's perfect for its utilization, as well as for crystal structures and different analysis. And so my process is to create like a predictive model that's able to compose all the different components that are needed in these cell-free reactions to make very specific membrane proteins. So the project kind of pulls in robotics, 3D printing, some microfluidics for the high throughput experimentation, some machine learning and protein structure modeling, and then obviously a lot of protein engineering and molecular biology. Yeah, and so with that, I just really want to thank sincerely the ARCS Foundation for all this funding. It's really freed up a lot of time for me, and I really appreciate it. So thank you so much.
Thank you, Connery. So our next caller is Deborah Park, PhD in pharmacology and toxicology.
Hi, it's nice to meet you all. I'm Deborah. I'm a sixth-year PhD candidate in pharmacology and toxicology. I'm working in Dr. Karen Zito's lab here at the UC Davis Center for Neuroscience. My thesis research is trying to understand the molecular and cellular mechanism that allows us to learn and form memories. And another part of my project is looking at how that same mechanism is potentially implicated in neurological or psychiatric disorders. Just to kind of briefly go into the specifics, I am looking at a protein called NN receptors that are present on dendritic spines. Spines are these little structural sites on brain cells that allow them to communicate with each other. And I'm looking at how the altered function of this protein, NN receptors on spines, are potentially implicated in the development of schizophrenia. This is a very debilitating disorder that affects millions worldwide. And hopefully the findings of my research can help further understanding of how schizophrenia develops, and also potentially expand upon therapeutic or treatment options for a lot of these symptoms are debilitating, ranging from cognitive deficits to hallucinations. And lastly, I'd like to thank the ARCS Foundation for the support. Also grateful to be a part of this community as well. And also this opportunity to share some of my research with you all today. Thank you.
Thank you, Deborah. Our next caller is Tyler Schleter, PhD in geology.
Hello. Thank you all for having me today. First off, I would like to express my gratitude to the ARCS Foundation for this award. I have been at UC Davis for 5 years now, and I have had to TA every quarter, with including summer quarters, with the exception of 2 quarters thanks to the ARCS Foundation. So I would be nowhere near as far on my dissertation research without the help that you guys provide. And so I will be eternally grateful for that. So as I mentioned, I'm a 5th year PhD candidate in the Earth and Planetary Sciences department. I study large silicic volcanic systems such as Yellowstone that are capable of producing catastrophic globe-altering eruptions. In case you didn't know, there's a strong hypothesis that about 75,000 years ago, an eruption in New Zealand known as the Toba eruption potentially bottlenecked the evolution of the human race. And so these events can have huge global effects. Specifically, what I do is I use novel microanalytical techniques in order to interrogate the crystal records extracted from deposits in New Zealand from the world's youngest super eruption, which occurred about 25,000 years ago. Crystal records are unique because they act a lot like tree rings in that they can record the changes in their chemical environment, and they can also be dated very well. So by using these data, I study the processes involved in the production and evolution of large silicic magmatic systems, specifically trying to understand how rapidly large volumes of mobile and therefore eruptible melt can be produced in the crust. My research has helped sort of further a paradigm shift in the field, excuse me, that has sort of demonstrated that these systems are able to reawaken and produce large volumes of melt on the timescale of decades to centuries, which was significantly faster than we previously thought, and therefore pose a significant hazard to humanity. So I hope to kind of parlay this information into sort of advocating for improved hazard mitigation efforts, as well as hazard assessment efforts. And yeah, that's kind of a rundown of my research, and I'm happy to talk about it more. And I just want to say thank you again for having me, and I'm excited to be here.
Thank you, Tyler. Our next speaker is Robert Tombery, PhD in chemistry.
Hello. Thank you for the introduction, and thank you all for being here and your extreme generosity. I'm in my 4th year working toward a PhD in chemistry with a focus on chemical biology. I work with Professor David Olson in a lab that studies synthetic organic chemistry, neuroscience, and a variety of topics in between. As a student with a chemical biology role in the lab, I design, synthesize, and test small molecule tools in a variety of assays to better understand the brain. More specifically, I work with molecules that are typically found in the brain, such as serotonin, and control their effects on various brain systems with light. As a group, we aim to combat and better understand neuropsychiatric diseases such as depression, addiction, and Alzheimer's disease, to name a few. I'd like to take this opportunity to thank the ARCS donors for your support and acknowledge that you are all fueling cutting-edge research that will leave a lasting impact on our world. In my breakout room, I'll be discussing a medicinal chemistry project where we designed a novel therapeutic that has anti-addictive and antidepressant properties. Thank you all.
Thank you, Robert. Our next scholar is Max Vargas, PhD in neuroscience, and I believe we have a video for Max.
That's where I shine. Give me a second.
Hello, everyone. My name is Max Vargas, and I'm a fourth-year neuroscience graduate student at UC Davis, working in the lab of David Olson. I'm fascinated by psychoactive drugs and how activation of their targets can ultimately lead to profound changes in behavior. This led to my interest in the molecular mechanisms through which psychedelics promote neuroplasticity, with the ability for the brain to rewire itself, and how these drugs can be used as therapeutics to treat depression, as well as other neuropsychiatric conditions. In recent years, psychedelics have been shown as fast-acting antidepressants, able to provide almost immediate relief to those suffering, while also providing long-lasting relief. And this effect is believed to stem from their ability to promote neuroplasticity in the neocortex, a brain region that experiences significant structural and functional detriments in depression. I focus on understanding how psychedelics and related compounds activate the serotonin 2A receptor. This particular receptor is the principal target of psychedelics and is necessary for both the hallucinogenic and plasticity promoting effects. My research lends evidence to the hypothesis that activation of intracellular serotonin 2A receptors by psychedelics is what is responsible for promoting neuroplasticity in cortical neurons. By researching this target, we can begin to understand the powerful effects of these drugs, and ultimately this can lead to the development of psychedelic-inspired therapeutics that can be safely used to treat depression and help those in need. And with that being said, I would like to thank the ARC Foundation for their support and funding in this research, and I would like to thank everyone in the audience for listening. Thank you.
Thank you, Max. So this concludes the introductions and short snippets from our scholars. I'd like to give them all a round of applause, a general round of applause for everyone.
Absolutely.
And like I mentioned before, we're going to be able to see them in breakout sessions. I want to pass the virtual mic to Leslie Van Houten for closing remarks.
Thank you so much. I will apologize if my dog barks. It's one of the joys of being home all the time. But I want to thank you so much for this amazing program. We give special thanks to Jessica Fowler-Ward and to Janet Berry. I understand Jessica did all the organizing and she did a great job. And so thank you both. And then to you, Dean Delaplanque, what a phenomenal group of ARCS scholars. Clearly you and the faculty deserve our gratitude for piquing your scholars' curiosity and nurturing them as young scientists. It's been so rewarding to learn the vast variety of work that they are doing and how they may affect our future in the years to come. It's just so exciting. And what an incredible group of ARCS scholars. We can't tell you how important it is for us to meet you one by one. And you are so inspirational, educational, and it is just an incredible opportunity for us. So thank you, scholars, for sharing your time and yourselves with us. We salute you and your academic achievements and your current and future contributions to science, math, and medicine. You are the future scientists of our nation, and we do know that we are in excellent hands. Thank you for sharing your journey. We are thrilled to be a very small part of that journey and help to contribute. Now we're about to go into our breakout session and we're randomly assigned. But at first I have to give a special thanks to 3 ARCS members who have designated scholars. In other words, they are totally supporting one of the UC Davis scholars. And they are Elizabeth Adams and her family, Jill Kramer, and Barbara Wolf. Now we'll be assigned randomly to our breakout sessions and look forward to more detailed involvement with our scholars. Thanks again to everyone who made this a very, very wonderful program for us.
Actually, I signed it up a little bit different, so let me explain it. So what I did is I assigned each one of our scholars to a room, and you'll see the name of the room with the ARC scholars. Now, our members, you guys get to choose which room you get to go to, and you can actually float around or bounce. So if you scroll over your screen towards the bottom, the same bar that has your mute and video functionality, you'll see a breakout room, and it looks like 4 squares. If you choose that, as soon as I push the rooms out, you get to choose your room, and you even can go back there to change rooms. Now you can send me a message or ask for help on the same bar, and I could jump in. For those of you that can't figure it out, or it just is not showing up correctly for you, I will randomly assign you. So I can help assist in the shuffling of sorts. Now my understanding is we don't come back, but the event ends at 11:30. And so thank you all for coming. And if you're ready, we can start the breakouts.